Hello, folks. It's a real pleasure to be here presenting to you at the Corteva Plant Breeding Symposium in West Africa. Now, today I'm going to be talking about predicting phenotypes and how we can use large data sets to accelerate both our understanding of plant genetics and predicting phenotype from genotype, but also plant breeding and predicting how new plant varieties will perform in previously unseen environments. So, as I'm sure many of you in this room are aware, our society, our civilization faces major challenges just in the next couple of coming decades. More than half of all the calories consumed around the world by human beings come directly or indirectly from three crops: maize, wheat, and rice. For two of those crops, wheat and rice, if you look at the countries and the regions where the yields are highest, yield stopped increasing sometime in the last couple of decades. This is obviously a major problem. A number of things have changed as a result. One is that after being relatively stable for quite a long period of time, the amount of land devoted to the production of grains, of foods generally, has had to start increasing again. But another is that world food prices have increased and stayed high. Many of you are probably familiar with the large price spikes we saw back in 2008 and 2011, but we are now back to almost the same all-time highs for food prices that we saw in 2011 in inflation-adjusted terms. Now, I mentioned three crops. I just told you about two. In maize, the situation is somewhat better. Yields are continuing to increase year after year. Since the mid-1950s, yields have been increasing at a rate of approximately 2 bushels per acre per year, incredibly consistently, to the point where this gets compared to Moore's Law, which was the prediction that the number of features on a CPU would double every 18 months. When you have a prediction like that, a very linear prediction of how much better products are going to get year after year, it really drives a lot of decision-making within industry, within plant breeding. Companies set their targets for how good new varieties have to be this many years out based on this projection of an additional 2 bushels per acre per year. But in order to achieve those increases, companies are having to spend more and more money on plant breeding. Now, there are a number of reasons for that. One is simply a basic law of diminishing returns, that it gets harder each year to increase yields because we've already selected for the easy to select for genes that have large effects and are at high minor allele frequencies in the population. But there's another factor as well. So it takes between 7 to 10 years in a conventional breeding program to develop and release a new variety. What I'm showing you here is an example from a wheat breeding program going through about 10 generations from generating an initial large population of F2s to actually releasing new varieties. In this breeding program, all successful plant breeding programs that I'm aware of, and in fact, those large increases in yield that we have seen for rice, for wheat, for corn, for many other crops, all depend on one critical ingredient, and that is selection in the target environment. I'm based in Lincoln, Nebraska in the US. I cannot use the yield data from here in Lincoln to predict which varieties will perform the best in another state in the US, like Michigan, or even another part of Nebraska. I have the yield data to show that if I pick my varieties based on Lincoln data and I try and select which varieties to grow in Western Nebraska, it does not work very well. So why is this such a hard thing? Because other traits are much more consistent. For example, if you look at flowering time between different environments, you can do a pretty good job of predicting which lines will be early flowering and which lines will be late flowering. Well, there are many factors at play, but one is that the more consistent traits like flowering time can have different effects on plant yield, on crop productivity, depending on the environment that they are in. What you see here is a very simple example from some yield trials we conducted here in Nebraska, looking at the yield of different corn varieties and the relationship between yield and flowering time. And what you see is that while it's possible to have low yield with any flowering time, The lines with the maximum yield potential really all flowered in this particular environment, Nebraska in 2020, between say 70 and 76 days after planting. In another environment with a different growing season, that will be different. These early flowering lines generally don't have time to make enough leaves before they go through the reproductive transition, so they don't fix enough carbon. These late flowering lines don't have enough time to fully mature before the frost comes and kills them. The environment isn't just the weather and the soil, though. It also represents agronomic practices. What you see here is a figure from a classical study looking at how different hybrids respond to different planting densities. And what you are seeing is that more recently developed hybrids produce the highest yield at very high planting densities, whereas earlier hybrids produced higher yields at moderate planting densities, and their yields actually went down if you planted too many of them together. They got stressed and were competing for resources. Again, we don't know all of the reasons for this change. We know this is something that plant breeders selected for. We know one of the features involved, though, is actually a change in leaf angle, with more recent hybrids having more erect leaves that distribute a smaller amount of sunlight, because each plant has a smaller share of the field over more photosynthetic area. This is one of the things that you really don't think about a lot until it changes. I had an opportunity a number of years ago to visit a plant breeding program in China, and the corn plants looked gorgeous, really happy, but really strange in some way that I couldn't put my finger on for quite a while, until I realized they still had these very flat leaves for the agronomic system that they were using and the way that they were growing the plants. That was the ideal for them. And so they had a set of genotypes that worked with those agronomic practices. The varieties I work with would not do well over there, and their varieties would not do well with the farmers that are my stakeholders at the University of Nebraska. But again, we have this problem, which is that all of our progress in breeding depends on selection in the target environment, and we no longer have access to our target environments, because if it takes 7 to 10 years to develop a new crop variety, a farmer's field in 2030 is going to be a very different environment from a farmer's field today. Temperatures are changing, growing seasons are changing. In the US, we're seeing changes in the timing and amount of rainfall that different parts of the country get. In addition to the climate, farmers' practices are also changing. So, for example, fertilizer is getting much more expensive. It's more expensive right now than it has been in the 21st century in the US. It's also likely to get more regulated, because state and municipal governments are getting concerned about the negative effects of excess fertilizer runoff, causing algal blooms, decreasing drinking water quality, and also just being extremely energy intensive to produce. So the future is an unknown environment. How do we develop crop varieties that will be able to succeed and thrive in those unknown environments? What we need are predictive models that are able to simulate how a particular variety of a crop may perform in a different environment, whether that is differences in rainfall or temperature, or differences in agronomic practices. You can envision, as I've just been talking about, how this would be beneficial to plant breeders, but it also is potentially beneficial to farmers. A farmer probably gets about 40 growing seasons in his or her adult life, where they're making the decisions about how, when to fertilize, what to plant, when to plant, what varieties to grow, whether or not to irrigate. And they have to be very conservative because if they make a mistake, they've lost one of those growing seasons and they're never going to get it back. So again, being able to simulate and say, well, if I did this, what would be the outcome, is a valuable decision support tool for growers. Now, I'm very fortunate to be part of the AI Institute for Resilient Agriculture, which is seeking to develop something called a digital twin, which is a in silico model of a particular crop plant that predicts how that plant will grow and respond to changes in its environment. These have been used very effectively in fields like manufacturing or transportation. So if you have a digital twin of a train engine, You can predict when individual parts are likely to fail and service them before the train breaks, but not have to service them until they're getting close to wearing out. Now I can talk at great length about how wonderful it would be to have these predictions, but the real question is, one, how do we actually develop them? And two, how do we convince people that the predictions are working? I'm going to focus more on the first one in this talk. The short answer is that we need lots of data on how plants perform in different environments. Now, this includes lots of collections of yield trial data and other conventional phenotypes, but it also means collecting data on phenotypes that would otherwise not be collected in a conventional breeding program. So these are the new technologies. These are things like sensors developed by my colleague Liang Dong, a very talented engineer at Iowa State University, that can lay on the surface of the leaf of a plant and measure how much water is being released by the plant as it photosynthesizes, how the humidity right by the leaf surface changes on different leaves and on different genotypes over time. It's also developed a nitrate sensor that can be inserted into the stalk of a corn plant and measure the amount of nitrate in the sap of the plant. Now, this is important because another one of my colleagues in the center, Mike Castellano, has shown that the nitrate concentration in corn stalks is a much better predictor than the nitrate concentration in the soil of whether or not it makes sense for farmers to apply more nitrogen fertilizer. If they're past a certain point, they're going to apply more fertilizer, but the cost of the fertilizer will be greater than the increase in yield that they get from adding that fertilizer. If the plant is nitrogen starved, though, adding even a little bit of nitrogen fertilizer will produce a lot more grain, and that's really good for the farmer. At the same time, oh, and I should mention I do have a conflict of interest. I'm involved in a company that's trying to commercialize these sensors beyond research uses. So what this shows you, though, is all of this data. We also have various types of robotics and UAVs and imaging. It all gets back to really predicting yield and predicting yield in different environments, like differences, different levels of soil nitrogen application and what that will do to yield and farmer profit. So can we do this at all? I have a postdoc in my lab, Ravi Moral, who was able to assemble a really large data set of about 160 different phenotypes that have all been scored for the same large population of crop plants. This is the SAM, it's two Maize association panels, and they've been grown in phenotype for different traits in different parts of the world. These traits have different heritability, so the proportion of the variance that is attributable to genetics. You can see flowering time is actually a pretty good one, with having a relatively high narrow sense heritability, which means we can do a pretty good job within a single environment of predicting flowering time based on genetic markers. For other types of traits can be more variable. So what Ravi and a PhD student in my lab, Michael Frost, did was a very simple question, which is that in the trials here in Lincoln, we had collected yield data from these same lines. And so they asked, well, how well can we predict yield using other phenotypes that we've measured from the same plots? And then secondly, which phenotypes of all the various ones we've measured for different experiments actually are important to the statistical models for predicting yield? They used an approach called a random forest model, which is relatively simple compared to something that would have like a convolutional neural network, but at the same time is a little bit more explicable, and it's easier to figure out which features are important to making the predictions. And it can capture nonlinear interactions, like I showed you with flowering time and yield. So what were the features that mattered? Oops, wrong way. Well, there were a number of features that came out as important in the model, but the three most important model features, the ones where if we randomized the data assignment for these particular features, the accuracy went down the most, are days to pollen. So one of our two flowering time measurements, because corn has days to pollen and days to silking, and these can be different. Plant height and the number of branches per tassel. Now, I would have predicted flowering time obviously was going to play a big role in determining yield. These others, I wouldn't have had strong feelings one way or another. But what's exciting to me about this is that all three of these, flowering time, plant height, and how many branches are on a tassel, are things that would be conceivably possible to measure with a UAV or other types of aerial imagery that we could fly over the field. So we can make predictions about yield before the end of the growing season. Corn is challenging for predicting yield otherwise because the ears tend to be buried down in the canopy, which is why we're working with various engineers who are developing robots that can drive through the canopy, although that gets much more challenging than the UAV or other aerial imaging. So how would we go about actually developing algorithms to measure things like branches per tassel from UAV data? I wish I had the slides showing you how we actually went about doing that. We haven't yet. This is a result that I saw for the first time Friday, so that's just yesterday. But I can tell you, yeah, none of these phenotypes came from high throughput phenotyping data. They came from the outstanding members of my research group who were out there walking through the field with rulers and notebooks using very old tech, but reliable tech for plant phenotyping. In order to develop high throughput methods to replicate some of what these people do in the field, we need a very complicated group of different expertises, and we need people with different expertise to work well together. Because I don't yet have that example of how would we go about, counting tassel branches from UAV data, I'm going to show you a simpler example of, again, how to go through these various steps and how we can start to learn about these different domains that are ultimately going to be things that we need to know as plant breeders and geneticists. So this simpler example is going to start with growth chamber data, or not greenhouse data, but controlled environment data. At the University of Nebraska, Lincoln, we have access to an automated greenhouse facility where plants move around on conveyor belts. They get weighed and watered every day. They come through a series of imaging chambers where they're photographed using different types of cameras. And this work is collaborations with one of my colleagues, Yufeng Ge, a very talented biological systems engineer who was hired about the same time that I was. Fortunate for me. So this system generates data that is in some ways easier to analyze. We're looking at the plant from the side, which means that we can see ears, not just the top of the canopy. We're also looking at individual plants against a contrasting background, which makes it much easier to separate out the plant from the background. This is from, this is almost, what, 4 or 5 years ago, 5-year-old data now. But it was incredibly valuable to my lab in learning how to do the basics of computer vision and processing images to extract different types of phenotypes. So what I'm going to show you here is some work from Jikai Liang, the first PhD student to join my lab. He's now doing a postdoc at University of Minnesota. So he came in, he was a molecular biologist. I handed him this data and said, okay, we need to figure out a way to analyze it, to measure phenotypes from these RGB images of corn plants. And he figured out how to do that. He figured out how to segment the image, look at things like the height of the corn plant. The R value here is quite good between ground truth measurements. One of the things I have learned is that when you start doing high throughput phenotyping, you end up having to do a lot more of the old-fashioned phenotyping as well, because now you have to have ground truth for everything to test how well your new technology is working. But you can see plant height works quite well. The ground truth measurements, the computer vision measurements are very correlated. For fresh weight, which he is estimating just from total pixel number, the correlation is still pretty good. The R value is 0.9, which means about 80% of the total variance can be explained by this relatively simple model from counting pixels of the plant from different viewing angles. But the really key question that Jikai asked was, what about all this error? I mean, the actual ground truth measurements are different from the predicted values. Is that random? If it's random, it's fine. We just collect more data and all of this random error will average out. It's not random. If it was random, I wouldn't be showing you the slide. Instead, it was systematic. What we found was that certain genotypes systematically had their biomass over or underestimated by our relatively simple algorithm for estimating plant biomass. In fact, 53% of the error in biomass estimates from images is the result of systematic over or underestimation of the biomass of specific genotypes. So if we used this simple algorithm, we'd used it to select lines to advance for, say, a biofuel breeding program, we'd be selecting for lines that had a lot of biomass, but also for lines that had traits that led to errors in biomass estimation. For example, differences in the ratio of leaves and stems, since leaves have less biomass per pixel. And so when Jakai had this result, and we speculated that this was something where it might actually be possible to map the genes controlling error in phenotyping. So, I mean, what are the genes that control error in phenotyping? Well, we have not done that for biomass. In a collaboration with the other Schnable lab, a research group at Iowa State University, they have in fact succeeded in doing this, looking at the genes controlling error in tassel phenotyping. This came out just earlier this year. So they were able to look at ground truth measurements of tassels and computer vision measurements of tassel, identify some cases where there was substantial error in either the length of the tassel or the length of the central spike. This tends to be correlated with whether it's an open or a sort of clumpy tassel. You can map genes for this openness. You can also map genes controlling the actual error, the delta between the manual measurement of the trait and the error of the trait. And I just think that's really cool. There are genes controlling error in our ability to measure the properties of plants. And that's one of the properties of plants. So how are we doing on time? Oh, we should be okay. In addition, I mean, so, you know, one approach, I mean, it's important to be aware of this as you start deploying high phenotyping technologies to guide your quantification of plant traits. But it's also important to think about, are there ways that we can approach imaging to reduce this challenge? So one is using new types of imaging technology that don't work like the human eye. What you see here is an example from sorghum, the other main crop we work on in my lab, where we're using a hyperspectral camera that is able to measure many, many different individual wavelengths of light. And using crowdsourcing or citizen science, we put these images up on a website where anyone could come and annotate the images and just tell us, you know, where are there leaves or stems or panicles? Where in this image are there pixels that are not plants. We very rapidly developed a pretty big data set of this, about 8,000 individually annotated pixels. From this, we could look at differences in the patterns of reflectance across both the visible and near infrared spectrum, and use these to train a computer vision, train an algorithm that was able to look at each pixel and classify it as belonging to one of these four categories, giving us these very nice images where we've conducted semantic segmentation. So not just, is this a plant pixel or not, but what type of plant pixel is each of these pixels? This is work from Chen-Yong Miao, another former PhD student in my lab, now working at Bayer. And Alejandro Pages, a computer science student, now employed by the University of Nebraska as professional computer scientist. By conducting semantic segmentation, we can get a little bit closer to the way that somebody, you know, a member of my lab or one of you might walk through a field and measure with a ruler and a notebook. By segmenting the whole plant and measuring the height from the bottom to the top, which is what you normally do with an RGB image, you get a lot of noise in the data because individual leaves come out of the top of a plant and then flop over. With semantic segmentation, we can measure to other landmarks like the top of the stem or the top of the panicle, and get something that is much more stable and monotonically increasing. I should probably speed up a little bit. This allows us to collect time series data, looking at how the height of different plants changes over time. Here in sorghum, we know 3 genes that control height, dwarf 1, dwarf 2, and dwarf 3. And by using this time series analysis, combined with functional principal component analysis, something that we learned about collaborating with a really talented statistician named Yuhang Xu, now at Bowling Green State University. We were able to decompose these time series data into functional principal components, conduct mapping for these individual functional principal components, identify the 3 genes that we knew we should be able to identify based on other studies, but which we couldn't with the relatively limited population that we were working with, as well as some additional genes that we hope to investigate in the future. Now I've shown you different traits that it's possible to measure from this semantically segmented image. What I haven't shown you is leaf angle, which I was talking about earlier is really one of the important traits in maize breeding as we bred to be able to pack more and more plants onto an acre in order to get more grain. The reason I haven't talked about leaf angle is that it's very hard to measure leaf angle from a 2D image. As the plant rotates, the apparent angle between the leaf and the stem will change, even though the true angle stays the same. So instead, what we need to do is be able to generate 3D reconstructions of crop plants. What you see here is work from the men's lab at Purdue University. Our collaborators working with some of the same image data collected from sorghum and using a voxel carving approach described in this paper to reconstruct the 3D structure of a sorghum plant from multiple 2D images. From this, they can conduct organ instance segmentation, so trying to identify which voxels, which 3D parts of the plant belong to which individual leaves, and then use this data to measure leaf angles in 3D. In fact, they get one angle for each leaf. Michael Tross, a PhD student in my lab, was able to use these leaf angles to conduct mapping for leaf angle in sorghum and identify both genes that we would expect to be identified as involved in leaf angle in sorghum because they've been identified in previous studies, but also some new hits that we have more confidence in because we also identify the things we expected to identify. So as we think about sort of the theme of this conference as a whole and how do we deploy new technologies to aid in plant breeding and ensuring food security, let's see. So obviously we can use in silico models to explore different combinations of genotype and environment and find new optimums that can be tested through plant breeding rather than trying to test everything in the field because there's just a limit to how much we can scale things in the field. In addition, as we try and scale to the field, we need to think about what are the technologies that really could be deployed at hundreds of field sites. Probably those will not be UAVs, probably those will not be ground-based robots. They're just both the cost of the equipment and the amount of personnel time it takes to keep the equipment running just doesn't make it feasible for a lot of breeding programs. However, it is now possible to buy satellite imagery with a resolution of about 30 centimeters a pixel, which means we get multiple pixels from a 2-row breeding plot of corn. I had an opportunity to collaborate with Symposium, a real expert on this type of data analysis. We showed that we can predict it's not great, but we can do a reasonable, a not terrible job of predicting the yield of individual yield test plots of corn using satellite imagery, which allows us to potentially bring some degree of time series phenotyping and high-throughput phenotyping to large numbers of different field sites throughout the US or throughout the world. I'm going to skip this one because we are almost out of time. The final thought I want to leave you with is yield per watt. So when we talk about yield, we almost always mean yield per acre, yield per hectare, how much grain or whatever crop you're growing can you get from a given field. But in many parts of the world, we are not limited by land. We're limited by some other reagent, whether that is nitrogen fertilizer, phosphorus, or even water. This is an example from Deepak Santra, my collaborator here at UNL. He works on a crop called prozone millet that gets used for a lot of the same things as corn or wheat or whatever. But it is able to produce more. I mean, the absolute yields are low, but the yields per unit water are substantially higher than that of corn. While the yields of corn have gone up in terms of per unit land, they have not gone up per unit water. So As we think about the future, it is probably not going to be just increasing the yields of our existing crops, but shifting our mix of crops in different parts of the world, depending on what is limiting in an area. Is it land? Is it fertilizer? Is it water? Is it something else? This will really determine which crops we use as the basis for keeping people fed. With that, I'd like to thank you all for your attention, and I understand I will be around in person to take questions a little bit later in the conference. Thank you.